Good morning, students. So this will be the second class on enzymology, and the class is on enzyme kinetics. So what is meant by kinetics? It is the study of rate of the reaction. Okay. So what is the function of enzyme? So I told you in the definition, enzyme will fasten the rate of the reaction, right? So in enzyme kinetics, we are going to learn how enzyme is going to fasten the rate of reaction and what are the factors which is going to affect the enzyme activity. So in enzyme kinetics, we are going to learn what is the mechanism of action of enzyme. That is how the enzyme is going to fasten the rate of the reaction and what are the factors which is going to affect the rate of the enzyme activity. So these 2 points we are going to learn in enzyme kinetics. So each of these are separate important 5-mark question. Okay, in mechanism of action of enzyme can be asked as a separate 5-mark question, and factors affecting rate of enzyme activity can be asked as a separate 10-mark question. So in today's class, we are going to learn about the mechanism of action of enzyme. So if this question is asked as a 5-mark question, I will tell you what are the points which we expect you to write in the exam. So the first point we expect is you should write down how an substrate get attached to the enzyme. That is how the substrate get attached to the enzyme to form enzyme substrate complex. First point. Second, what is mean by activation energy? What is mean by activation energy? Okay. Third, how enzyme is going to reduce the activation energy. What are the mechanism by which enzyme is going to reduce the activation energy? So these are the 3 points which we expect you to write when the question is asked as a mechanism of action of enzyme. Okay, let's see one by one. So the first point is you are going to explain how the enzyme is going to get attached to the substrate to form enzyme substrate complex, or else how the substrate is going to get attached to the enzyme to form enzyme substrate complex. Okay, this is explained by 3 different theories. The first theory is called as Michael-Menden theory. Michael-Menden theory. Second theory is called as Fisher template theory. Fisher template theory, otherwise called as lock and key theory. Okay. Second theory is called as Fisher template theory or lock and key theory. And third one is called as Koestlander induced fit theory. Okay, Koestlander induced fit theory. So these are the 3 theories which explain how one substrate will get attached to enzyme, result in formation of enzyme substrate complex. Let's see one by one. Okay, we'll start with the Michael-Menden theory. So according to this Michael-Menden theory, so it was explained by Michael and Menden in 1913, and according to this theory, that is according to them, we have an enzyme, we have a substrate. Okay, so this is the active site of the enzyme. Okay, and this substrate will get attached to the active site of the enzyme and it will get converted to product and it will be released from the enzyme. Okay, so there are totally 3 steps. What is the first step? You have enzyme and substrate. Okay, second step, the substrate will get attached to the enzyme to form enzyme substrate complex. Third step, the substrate will get converted to form product and the product will be released. Okay, so these are the 3 steps which was explained by Michael-Menden theory. Okay, but the problem with this theory is in definition we had learned enzymes are very specific in their action, but here the theory cannot explain how an enzyme, that is according to this theory, any substrate can get attached to the active site of any enzyme, right? So here they had explained you have a substrate and enzyme, then you have enzyme substrate complex, then product and enzyme, right? But this theory does not explain how one particular enzyme is specific for one particular substrate. Okay, it does not explain that specificity of the enzyme. Okay, it just explained you have enzyme and substrate and then enzyme substrate complex, then a product and enzyme is formed, the product is released. Okay, so they had explained we have 3 steps. Okay, but the limitation is it does not explain the specificity. Okay, it was explained by Michael and Menden in 1913. Okay, so we have 3 steps. So this limitation was overcome by next theory, which is called as Fisher template theory. Okay, according to this Fisher template theory, he explained that the active site of the enzyme is rigid. Okay, the active site of the enzyme, this is the enzyme and this is the active site of the enzyme, it is rigid. Okay, and he says that the active site shape is complementary to that of the substrate. Okay, if you have 2 substrate, okay, only this substrate can get attached to the active site because the shape of this particular substrate is complementary to that of the active site of the enzyme. Okay, this substrate cannot get attached to the active site because the shape of this particular substrate is not complementary to that of the active site of the enzyme. Okay, is it clear? So the limitation which we are seeing in the first theory is overcome by Fisher template theory. Okay, according to this theory, the active site is rigid and it is complementary to that of the substrate. But there is a limitation in this theory also. For example, let's take the last step of anaerobic glycolysis, that is conversion of pyruvate to lactate by lactate dehydrogenase. It's a reversible reaction. So the same enzyme can able to convert pyruvate to lactate, also lactate to pyruvate. Okay, so according to this theory, the substrate should be complementary to that of the active site of the enzyme. Okay, let's assume pyruvate is a substrate here. So the active site is complementary to that of the pyruvate. So pyruvate will get attached to the active site. Okay, so this theory explains the specificity of the enzyme. Okay, so only those substrate which is complementary to that of the active site can get attached to the active site. That's what this theory explains, right? So if pyruvate is complementary to that of the active site, it will get attached to the active site, it will get converted to form product, that is lactate, and the lactate will be released. Okay, but lactate cannot get attached to the active site according to this theory. But in reality, what is happening? Lactate also can get attached to the lactate also can get attached to the lactate dehydrogenase enzyme. It will get converted to form pyruvate. Okay, so the advantage of this theory, it explains the specificity of the enzyme. Okay, but the disadvantage is when you have a reversible reaction, this theory cannot explain how a substrate and product which is structurally different can get attached to the same active site. Okay, so this is the limitation of Fisher template theory. So it was explained by Fisher. The active site is rigid and pre-shaped, and the active site will be complementary to that of the substrate. So it's like a lock and key. Okay, because only one particular key can able to fit into one particular lock. Okay, that's why it is called as lock and key. Lock is like the enzyme, key is the substrate. Okay, only one particular substrate can get attached to the active site of the lock, that is enzyme. Okay, that's why it is called as, otherwise called as lock and key theory. Fisher template theory is otherwise called as lock and key theory. Okay, but so it can able to explain the specificity of the enzyme, but it cannot explain how an in reversible reaction a substrate and product can get attached to the same enzyme. Okay, so this was overcome by next theory is Koechlander induced fit theory. According to this theory, the active site of the enzyme is not rigid. It is flexible. Okay, this is the active site of the enzyme. This is the enzyme. This active site of the enzyme, according to this theory, it is not rigid. It is rather flexible. When the substrate comes in contact with the active site of the enzyme, the substrate will induce an confirmatory changes in the active site. Okay, in such a way that the active site of the enzyme will undergo confirmatory changes and it will become complementary to that of the substrate. Now the substrate will get attached to the active site. It will get converted to form product and the product will be released. Okay. So the active site is not initially complementary to that of the substrate. Once the substrate comes in contact with the active site, the substrate will induce confirmatory changes in the active site in such a way that the active site of the enzyme, now it will become complementary to that of the substrate. Now the substrate will get attached to the active site and it will get converted to form product. Okay, so this theory can able to explain how the substrate and the product in a reversible reaction which are structurally different can get attached to the same active site of an enzyme. Okay, so Koechlander induced fit theory explains that the active site is not fixed, it is rather flexible. Okay, so when the substrate comes in contact with the active site, it is going to induce in confirmatory changes so that now the active site will become complementary to that of the substrate, and now the substrate will get attached to the active site and it will become product. So we have seen about three theories which explain how the substrate will get attached to the enzyme, result in formation of enzyme substrate complex. Next, coming to the activation energy. Let me explain the activation energy with a simple diagram. Okay, let's assume I'm going to give you a stick and I'm going to ask you to break the stick. So what you'll do, you'll hold the stick with your both the hand and you're going to apply pressure over the stick. Okay, when you apply pressure over the stick will bend, that is it will go to a transition state. Okay, after that it will break. Okay, that is you are applying, holding it with both the hand, you are going to apply pressure over the stick, that is you are applying energy over the stick so that it will bend, it will go to a transition state. This is the transition state, then it will break. Okay, so instead of this you are going to replace it with substrate. Instead of this image, you are going to replace it with the product. Okay, so you are taking the substrate and you are going to provide energy. Okay, at one particular point, the substrate will reach a transition state. Okay, the substrate will reach a transition state. Okay, and after that it will immediately get converted to form product. Okay, so we are taking the substrate and we are providing energy. So the substrate is going to reach the transition state, then it will get converted to form product. Let me explain the same thing with the reaction coordinate graph. So what is a reaction coordinate graph? Here I'm going to draw a graph with energy which is provided to the substrate along the y-axis and the conversion of substrate to the product, that is chemical reaction, along the x-axis. Okay, so this is the substrate which is present in the ground state. Now I'm going to provide energy to the substrate so that the substrate will reach a transition state. Once the substrate has reached this transition state, it will get automatically get converted to form product. Okay, so this is the transition state. Okay, so the amount of energy which is provided to the substrate so that it will move from the ground state to the transition state is called as activation energy. So what is activation energy? It is the amount of energy provided to the substrate so that the substrate will move from the ground state to the transition state. Okay, so this is the activation energy for this particular reaction to convert the substrate to form product in the absence of enzyme. Okay, this is the activation energy in the absence of enzyme. Okay, let's see what happens in the presence of enzyme. The graph will appear like this. So in the presence of enzyme, the graph will be like this. So what has happened now? The activation energy is reduced. The activation energy is reduced. So in a single sentence, you want to say what is the mechanism of action of enzyme? The enzyme reduces the activation energy. Okay, the enzyme reduces the activation energy. Okay, so more number of substrate will get converted to form products, so the rate of the reaction will be increased. Okay, so if you want to understand this in a very simple way, let me explain this with some other example. Okay, let's assume in university exam, the university has decided to increase your pass cutoff. Okay, so only if you get 90% in your final year university theory paper, you will be given, that is, you can able to pass the first year. Okay, so what if the cutoff is increased to 90%, from 50 to 90%, the number of students who is going to pass the first year will reduce, right? Only 10 or 5 students will pass the first year. Okay, let's assume another scenario where the university has decided to reduce the pass percentage, okay, to 10%. Okay, any students who has crossed 10% in the theory exam, they are allowed to enter the second year. Okay, so then what will be the result? All of the students will be passing the first year, right? So by reducing the cutoff, we are increasing the number of pass percentage. So by reducing the activation energy, we are increasing the conversion of substrate to the product formation. Okay, that is, we are increasing the rate of the reaction. Okay, this is how enzyme increase the rate of the reaction. That is, it reduces the activation energy. So what is activation energy? Activation energy is the energy required to convert all molecules in 1 mole of reactant substance from ground state to transition state. Okay, so what does enzyme does? Enzyme is going to reduce the activation energy. Okay, thereby it is going to increase the rate of the reaction. So we have seen how the substrate will get attached to the enzyme to form enzyme substrate complex. The first point. Second, we have seen what is meant by activation energy, and I have told you enzyme will reduce the activation energy. So the third point, you are going to explain how the enzyme is going to reduce the activation energy. What are the mechanism by which the enzyme is going to reduce the activation energy? So at least you should be able to explain this 4 points. That is, these are the 4 methods by which enzyme will reduce the activation energy. The first point is substrate strain. Okay, second is covalent catalysis. Third is acid-base catalysis. Fourth is entropic effect. Okay, so these are the 4 important points which we are going to see now. Okay, let's start with the substrate strain. So what is substrate strain? Okay, let's assume you have a substrate and you have to put a pressure over the substrate so that the substrate will get broken to form 2 product. Okay, let's assume you have a scenario like this. Okay, so for that I have to provide an external energy. Okay, I have to provide energy from external so that I can able to break this substrate to form 2 products. Okay, instead of that, what can I do? I will place the substrate in the active site and the enzyme will put a pressure over the substrate. That is, it is putting a strain over the substrate in such a way that substrate will get broken down and it will form product and the product will be released. Okay, so that external energy which is required to break the substrate to form product is saved. Okay, so we are reducing the activation energy. Okay, this is how we are going to reduce the activation energy by substrate strain. That is, the active site of the enzyme is going to put a pressure. It is going to put a strain over the substrate. So the substrate will become product. This is called a substrate strain. Okay, so that we are going to save the energy. We are going to reduce the activation energy. Second, covalent catalysis. So what is covalent catalysis? When substrate get attached to the active site of the enzyme, multiple covalent bonds will be formed between the substrate and the enzyme. Then the substrate will get converted to form product and the product will be released. Okay, the product will be released. Now you will be having a question, how by just forming covalent bond with the substrate we can able to save energy. Okay, so this you can able to understand with a simple example. Okay, let's assume you have a pearl necklace. Okay, let's assume you have a pearl necklace and let's assume it is very slippery. Okay, my aim is to cut the thread between the pearl using a scissor. Okay, my aim is to cut the thread between the pearl using the scissor. Okay, and let's assume it's a very slippery pearl necklace. Okay, so when you try to cut the thread between the pearl without holding the pearl necklace, it will be difficult for you. You have to make multiple attempts. Okay, it will slipping, right? Multiple time it will slip. So I have to make multiple attempt to find out the thread which is present between the pearls. Okay, but what if I hold the pearl necklace with two hands and if I stretch it, then if I try to find out the thread between the pearl and cut it using the scissor, now I can able to cut it with a single attempt itself, right? So that multiple attempt is saved. That is the multiple time I am trying to cut this thread is saved. So we are saving the energy, right? So like this, by holding the substrate at that particular place in the active site, we are saving the energy by holding it in the particular place. Okay, that is we are holding the substrate in the particular place so that we can able to cut this substrate in a single attempt. Okay, we can able to convert the substrate to form product in a single attempt. Okay, so that we can able to save that energy. Okay, this is called as covalent catalysis. Next, coming to acid base catalysis. So what is meant by acid base catalysis? Let's assume this is the substrate and this is the active site. Okay, let's assume this substrate has an extra hydrogen. If I remove that hydrogen from the substrate will become product. Okay, so that removed hydrogen has to be taken up by some other molecule. Okay, and that will be taken up by the base which is present, the basic amino acid present in the active site. Okay, instead of an external molecule which is going to consume that hydrogen which require energy, here we are having basic amino acid in the active site that is going to taken up the hydrogen from the substrate so that the substrate will become product. Okay, in another scenario you have a substrate which require hydrogen to get converted to form product. Okay, in that scenario the acidic amino acid which is present at the active site is going to donate the hydrogen ion so that the substrate will become product. Okay, so the acidic amino acid will provide a hydrogen ion to substrate to become product, and the basic amino acid will accept the hydrogen ion from the substrate to become product. Okay, so by this method, acidic and basic amino acid which is present in the active site is going to involve in the transfer of hydrogen ion to the substrate, result in converting the substrate to the form product. Okay, so that the external source is not needed to provide or accept that hydrogen ion, so that energy is saved. Okay, this is called as acid base catalysis. Okay, that's why if you, when you see in active site, when you read about the active site in your book, they will mention that the active site is mainly made up of acidic and basic amino acid because it is involved in acid base catalysis. Okay, acid base catalysis. So this is about acid base catalysis. So now we have seen about substrate strain, covalent catalysis and acid base catalysis. Next, coming to the entropic effect. So what is entropy? Entropy is the disorder in a system. Okay, for example, let's say you have substrate A and substrate B. Okay, and you have to combine these two substrate to form a product. Okay, and let's assume the energy which is required to combine substrate A and substrate B to form product is, let's assume it's 50 kilojoule. Okay, and in reality, instead of 50 kilojoule, you will be consuming 70 kilojoule to combine this A and B to form a product. Okay, so that extra 20 kilojoule is called as disorder in the system. That extra wastage of energy is called as disorder in the system. Okay, let me explain this with a very simple example so that you can be able to understand. Okay, let's assume, let's take a scenario in which your mother has asked you to buy half a liter of oil. Let's assume the market price of the oil is 50 rupees and your mother has given 100 rupees. okay, and she has told you to go to the shop and buy half a liter of oil. You went to the shop, okay, and you bought half a liter of oil that cost around 50 rupees. Okay, now tell me whether you will buy only oil or else you will buy some snacks for you also. You will buy some snacks for you also, right? So that will cost around extra 10 to 20 rupees. So instead of spending 50 rupees for half a liter of oil, extra you had bought some snacks which is around 10 to 20 rupees extra. So you had expended 70 rupees. The extra 20 rupees is the disorder in the system. Okay, so that can be avoided by enzyme. Okay, so if you take another scenario, in this scenario your mother is going to come along with you to the shop to buy the oil. Okay, now you cannot spend that excess amount of money for buying the snacks. Okay, now you can able to spend only that 50 rupees to buy half a liter of oil. Okay, that is enzyme. Okay, so enzyme will reduce the disorder in the system so that it will reduce the requirement of excess amount of energy. Okay, so how it is going to reduce the disorder in the system? There are two methods. Okay, let's see the two methods. The first method is called as catalysis by proximity. Okay, let's assume this is substrate A and this is substrate B. Okay, let's assume to combine the substrate A and B to form a product, I have to drag this substrate A to here and substrate B to here, and I have to rotate it in such a way that this red part of the substrate A should come in contact with the blue part of the substrate B. Then only it will get converted to form product. Okay, so to drag this substrate A to this position and substrate B to this position and to rotate it in such a way so that this red part and blue part will face each other, I need to spend some amount of energy, right? So that's the disorder in the system. So this can be prevented by enzyme. So what enzyme will do? The enzyme will attract the substrate A and substrate B towards the active site. Okay, it will be attracted and attached to the active site in such a way that the red part of the substrate A will be facing the blue part of the substrate B. So these will be interacting with each other to form the product and the product will be released. So the amount of energy which is required to drag substrate A and substrate B and to rotate it to form product is not needed. So that is saved. So that particular wastage of energy is saved. Okay. So by this means we can able to reduce this entropic effect. Okay. Second method is called as desolvation. Okay, if you see in our body, in blood, which is made up of mainly made up of water, all the molecule which is present in the blood, that is present in the water, will be surrounded by a water shell. Okay, so let's assume this is substrate A and this is substrate B, and it is surrounded by water shell. Okay, if I want to combine the substrate A and substrate B, I need to remove this water shell first. Okay, then only I can able to combine these two to form product. Okay, so to remove this water shell, I need to spend some amount of energy. That action will be done by the enzyme. So enzyme will remove the water shell so that the substrate A and substrate B will combine to form product and that will be released. Okay, so that energy which is required to remove the water shell will be done by the enzyme. So that energy is saved. Okay, that is one of the entropic effect. Okay, so what are the mechanism by which the enzyme is going to reduce the activation energy? By substrate strain, covalent catalysis, acid-base catalysis, and entropic effect. Okay, so these are the four methods. So what we had seen in the mechanism of action of enzyme, first we had seen how the substrate will get attached to the active site of the enzyme to form enzyme substrate complex. Then we had seen what is meant by activation energy, and we have seen enzyme will reduce the activation energy. And finally, we had seen what are the mechanism by which enzyme is going to reduce the activation energy. Okay, that's all about mechanism of action of enzyme. Thank you.